first speaker on this session dealing with CRISPR-Cas9 and other genome editing tools is Dr. Fayaz Khazi. He's Chief Executive Officer of ELO Life Systems, based in the US. It's an agriculture company that enables, through partners in food and ag industry, to meet the needs across agricultural productivity, nutritional demand, food security, and human wellness through rapid deployment of technology, and you couldn't want anything more rapid than genome editing. So if I've missed anything supportive out of you, out of my introduction, Fayaz, please introduce that. And your title is also new, which is Translational Genome Editing and Beyond. Fayaz, you have the floor.
Thanks again. Thanks for the invitation. I don't know if Maurice is in the audience, but I'm honored to be here and talk a little bit about ELO Life Systems. We went back and forth. When you have ELO, it stands for Electric Light Orchestra. But ELO, with E in caps and the other two letters in lowercase, rhymes with hello, or hello in Australian. It basically stands for the world in Cherokee, in Native American language. It's the world. So we were looking for something that kind of spoke about what we wanted to do using genome editing and other technologies, which was to address things at a systems level. And therefore, we kind of thought, ELO was an appropriate name for the company. Okay, so I was assuming I'll have maybe 20 minutes, so I put 5 slides in one slide and said, this is my background slide, but it looks like I may have to elaborate further here. So the first few slides, this is how I organized the presentation. The first few slides, I'm going to walk you through what the technology is and how we came about and how long have we been around and those kind of stuff. And then the next part of the conversation is, why do we think focus on human health and connecting the gap between agriculture, what we eat and how we live is essential. And then in the third part, we're going to put those two things together and talk about how exactly are we doing it? How are we innovating in our space? And how we would like to convert innovation into adoption and then with a maximum impact. So that's the idea. So speaking of genome editing technologies, this technology or this system, the natural genome editing system, which is based on homing endonucleases, has been around for a very long time. This is Chlamydomonas reinhardti, and everybody's hopefully familiar with the ICRE1 homing endonuclease system, which the algae uses. It's a self-splicing intron, and the couple of things that are cool about this is, number one, it is conserved. It's, this is the quintessential definition of a selfish gene or a selfish piece of DNA. It's all about itself and nothing else. So in here, a couple of things that resonated with the community that was looking at ICRE homing endonucleases early on, where, number one, it has a very slow catalytic rate. So it binds and then it cuts, but it takes its own time. And all those things become relevant as we talk about a product-grade genome editing tool. The second part was, no turnover. That means the nucleus remains bound to the DNA end. So the nucleus gets in, identifies a cognate site, and then there is a catalytic site that cuts the DNA. And once that DNA is cut, the nucleus still remains there. It doesn't run away as quickly as other DNA repair proteins do to go and repair, cut and repair something else. So it's in all sorts. It's more of a really lazy system. And then the third one, the active site, it does not form until the enzyme has bound productively to the correct DNA site. That means there is a 3-dimensional component involved in the DNA-protein interaction. Unless it integrates or binds covalently to the DNA sequence, it cannot cut. So it has to bind first. First it has to recognize, then it has to bind, and then it forms a different conformation, a three-dimensional conformation, which allows it to cut that side. So the next part is far more interesting than the first three things here. The pattern of the cut. So it recognizes a 22 base pair target site. And that 22 base pair target site, if you look at the size of the genome, in most cases you are able to find a unique sequence that's 22 base pair long in any eukaryotic genomes. And then the next part is the kind of a cut that it generates. It generates a 4 base pair overhang, a 3' overhang, like here. So this essentially allows for it to initiate homologous recombination activity. And by this, people, when we have studied homologous recombination, there is strand invasion, and then there is strand exchange that's followed by the strand invasion. So this is critical as we start talking about genome editing and the control on the editing process itself. What's been happening here is, the iCRE system has evolved to create a overhang, and the overhang has the ability to invade a homologous strand, and therefore it propagates itself. So it was essential for the iCRE nuclease to have all these characteristics so it can selfishly replicate as the chlamydia1 has replicated. Yeah, so that's something to keep in the back of your mind as we talk about applications. So we looked at, in the early days, about a decade ago, the original founders of Precision Biosciences, who owns this technology, and Elo is a subsidiary of Precision focused on food and agriculture, looked at different families of homing endonucleases found across multiple kingdoms. And based on that, we because most of the information that was available was iCRE1 related, so we did the more practical thing. We started looking at iCRE1 in more detail and started building a system that was based on this naturally occurring genome editing platform. So initially, basically we took this together, and then we identified the bridging contacts and made those bridging contacts interchangeable. So we identified those regions which were absolutely essential, meaning those essential regions, if you disrupted them, it would lose its characteristics as a homing endonuclease. So we retained those, but then those interchangeable regions, we characterized them further to pretty much control the DNA binding affinity, right? So then the next part was active site configuration. Once again, looking at the amino acid configurations and a good amount of three-dimensional modeling, we were able to identify not only improve the catalytic kinetics, but also able to make this more of an inducible system, meaning it needed, so we connected the binding and cutting as closely as we could in a synthetic system that is based off of iCRE1. And then finally, the whole protein DNA interface we were able to redesign, and we had to redesign this because the naturally occurring iCRE1, it was a rigid, slow-moving, slow-acting enzyme. For us to be able to have a platform that was product grade, we needed it to be reliable, rugged, robust, and reproducible. So for those reasons, we were able to make all these changes here. And then we ended up with what we call as ARCUS. ARCUS is a single gene, meganuclease-based enzyme that essentially is less than a kb in size. So, and the size in itself matters quite a bit. So when you put this against, the current technologies that are out there, there are several features that stand out. This is your old school ICRE1 meganuclease. We are looking at ZFNs here, TALENs, and CRISPRs. So when compared to all of this, the things that stand out about ARCUS are it's a platform that produces a small, scalable, proprietary nucleases that coordinate cutting and binding to ensure specificity and can be customized to target any genes. So whereas in here we talked about all the things that really don't work about ICRE1 as a product-ready genome editing system. ZFNs, for example, they use a non-specific nuclease. So the DNA binding domain's domain. So every time a ZFN pauses somewhere, the nuclease is ready to chop off the DNA. And the similar case with TALENs as well. CRISPR, I think in this case, while CRISPR technology is robust, the Cas9 protein is massive, and it also creates challenges in relation to delivery. So over the last 9 years or so, we've spent, staring at about close to 6,000 in silico models of ARCUS nucleases. A whole bunch of nucleases have been tested, and more than 1 million. I don't know how people get to calculate those, but that's what I was told. More than 1 million enzyme digests. What's important here, once again, from a product-ready enzymatic or a genome editing platform perspective, number of patent lawsuits defended, 3. So this is, and we were successfully able to defend the patents and the technology in the court, and we established freedom to operate. And over the course of 9 years, we've spent about $20 million, which doesn't seem like a lot when you really think about, what we got and how much time we spent on this one. Okay, so to summarize this, what we have is, now that you have a product-ready platform, what does a product-ready platform look like? And how can you take a genome editing system from, from a concept to finally to a product? So there are a couple of technical things that need to be set right. So number one is delivery. Delivery is pretty straightforward with an ARCUS nuclease compared to any other genome editing technology that's out there, because it's only less than a kb in size. And it's a single chain nucleus molecule, and it fits into pretty much any delivery system that's out there. That cannot be, the same cannot be said for any other genome editing platforms today. The next one is targeting. So in here, because we are able to, we have the hypervariable regions within the protein, which we can modify based on the target. We are able to pretty much target any part of the genome. So that makes it quite a robust technology for us to, generate products in a variety of systems, that basically any system that has DNA in it. The repair part becomes really unique because initially, when we all started thinking about genome editing, it was essentially knockouts. The next level of genome editing is going to be, targeted deletions, targeted insertions. And down the road, it's going to be, homologous recombination-based specific insertions and specific modifications with cisgenic genes and whatnot. So for all those things, right now there is a huge amount of effort that's being spent in the industry on understanding, defining, redefining HDR, homology-dependent repair. In this case, we have a leg up, basically because the kind of cut the enzyme does by default is a 3' overhang. It creates a situation for homology-dependent repair. And, oh, there we go. And efficacy. Efficacy is a function of both delivery, targeting, and repair, all three. And in this scenario, the proprietary binding and active site mutations that we have basically give you the control over targeting the right sequence, not targeting the wrong sequence, and cleavage, the right kind of cleavage at the right kind of targeting sequence. So based on this, the efficacy, in my opinion, is pretty much covered. And that part leads to the safety aspects of it. So to date, we have 0 random offsite targeting. And in certain situations when we come across more offsite targeting than we can tolerate for any product instance, we are able to go back to the drawing board and evolve an enzyme, recreate the enzyme that has 0 offsite targeting. So that, right now, you know, two of our clinical programs will be online next quarter and early in 2019. This is going into people for CAR T cell-based program and also for gene therapy programs. And for plants, especially when you're looking at clonally propagated crops, where, you know, germplasms have been nurtured over, you know, the last 5-10 decades, you really want a system that does not perturb anything else but the target gene, yeah, or the target sequence. So for all those reasons, I think the ARCUS platform is pretty much ready for prime time. And what we did was, over the last 10 years or so, we've been partnering with, you know, pretty much all the big ag that was there and which is merging at this point. You know, we pretty much work with everyone, and we published this in good journals. And arguably, as far as I know, the male sterile maize plant that we created in collaboration with Pioneer Hybrid was the first ever genome edited crop plant to be reported. So at this point, we are testing out, you know, a variety of systems. We are looking at multiple delivery systems, and also using these delivery systems, taking advantage of the small size of the nuclease to deliver multiple nucleases at the same time to address metabolic pathways. So there are partnerships that we can talk about, and then there are partnerships that we cannot talk about. So the partnership that we can talk about, this is basically a snapshot from Cargill's press release, and this is one of the ones that we are very proud of. This is the program where we collaborated and partnered with Cargill to develop ultra-low saturates in high oleic canola oil. This press release came out sometime in February, and the plan is, right now these things are, the plants are in the field, and it's going into the next stage of initial commercialization. Discussions are ongoing. So this is time to switch gears a little bit, right? So we talked about applications in your standard crop plants. Then we also talked about partnerships with companies in the business who have the, you know, vertically integrated pipeline to start from genetics and take it to product processing and product. So the next part is, you know, want to focus on who we are as a company and where we are headed as a company. Okay. So in this part, we are a company as a whole, Precision and Elo, focused on human health and wellness. The Precision arm, the mother company, is focused on curing diseases. It's focused on genetic and infectious diseases, whereas we are focused more on you know, introducing healthy alternatives to the market. So both, we are looking at different phases of human health. Yeah. So Precision at this point, we've been in business for, like I said, around 12 years now. We are focused on hemophilia A, hepatitis B, retinitis pigmentosa, and familial hypercholesterolemia, and also a couple of their cancer immunotherapy projects, which are, so one of the cancer immunotherapy programs is going to go into the clinic, and one of the gene therapy programs, I think more likely, is the familial hypercholesterolemia that's going to go into the clinic early next year. As far as Elo is concerned, like I said, we are a food and agriculture subsidiary of Precision. We basically announced the new name maybe 3 weeks ago, so brand new. This is my first presentation with Elo logo on the slide there. So what we are focusing on are, introducing healthy alternatives to the market. By that, what we mean are, we are looking at really serious and some might call boring things, but things that impact everyone. So, the first start has been the ultra-low saturate oils. The next one we are looking at also are low-calorie natural sweeteners. And improved protein profiles in various crops, improved nutritional profiles in various crops. And then we are also working with entities to expedite adoption of emerging crops to the market. And all of this is coming from, feedback we receive from our partners. So we are not betting on traits in a vacuum. We are not betting on traits because my best scientist said this is something cool, this is what we need to do. What we are trying to do is identify what is preventing the food industry, the beverage industry, the seed industry from holding back and not introducing a diverse array of food products on the grocery shelves. And the common answer that we receive is either things don't fit into their processing pipeline, or there are no products that consumer likes because of texture, flavor, color, whatnot. So that's the kind of feedback that we are basing our strategies on, and we will be moving forward along those lines. And the hallmark of everything is we are very translationally focused. So when we start a program, we have an end user in mind, we have a final customer in mind, we have a product developer in mind and in the discussion for that program. So none of these are model planned, none of these are fun to do kind of experiments, but these are all purpose-driven experiments. And then the last thing is elaborated further in this slide, which is the business model in itself. For a company the size of ours, at this point, Precision has about 110 people on staff, and Elo is only about 12 people. I have 16 or 20 positions to fill in the next 3 months. So we will double in size by the end of this year, hopefully. But the approach we are taking and the partners we already have are pretty significant. So, like I said, everything is going to be industry partner initiated ideas, and then it will be guided by the customers, and the technology development is going to happen here. And the idea here in terms of rapid innovation is multi-scale biology-based knowledge generation. So we are tapping into all the different components of innovation, starting from idea concept generation, all the way to partnership identification, and then taking it into the market. So at this stage, what I like about the multi-scale biology approach is from the get-go, we are looking at not just one crop as it stands on its own, but the relationship between crops. Things that are true for pigeon pea could be true for chickpea and could be true for other lentils. So we would want to build a pipeline where we don't have to go back and start from scratch on every crop. So we are staging and we are building relationships where there is a strong understanding that, if we work on chickpea, then the same partners will be able to move other lentils also forward along the lines of chickpea, for example. Yep. So, and then finally, this design, build, test, and partnership-based iterative cycle is going to become pretty robust, as you can tell. And all of this is our cornerstone technology is the genome editing technology. And not for a single day we believe that genome editing is going to solve all the complexities in agriculture. So that's why this is what gets the attention. FTO brings the people, the partnerships, and the money. But we are sane enough to start identifying and already identified a whole bunch of enabling technologies that will make us take the ideas to products. And then here's a illustration that talks about the 4 units within ELO. One is knowledge management, which is basically multi-scale biology, computational biology, gene discovery. The second one is technology development, which is not just Arcus nucleus technology development, but also looking at gene delivery technologies and multiplexing technologies and high throughput screening and validation technologies, protoplast transformation technologies. The third one is the translational agriculture team, where this is the group that interfaces with partners and identifies marketable, translatable ideas, identifies, issues with the thought process that we have with one or the other project, and they are basically the duct tape of the company. And then the tissue culture and controlled environment agriculture is the foundational infrastructure that supports all the activities through the design, build, test, and product development phase. And on the right-hand side are all the sector partnerships. We don't have partnerships in animals, aquaculture, fuel at this point, but for the others, we do have partnerships already in place. And the kind of agreements that we enter into are called pacts. The partnerships, these are partnerships advancing crops through technology. So that's the idea, and I think we've been receiving pretty good support from the industry and academia and not-for-profit organizations at this point. So in terms of growth, we recently signed a deal with Alexandria Realty Equities, which is a company that has multiple incubators and R&D sites all over the country in the US, in Boston, in San Diego, in San Francisco, and also now in RTP. What we are trying to build here in collaboration with these guys is an infrastructure, a foundational infrastructure for ag-related research. And this building used to be the old Syngenta building. I was just talking to you about that. And it doesn't look like the old Syngenta building anymore. Right now, it's just a parking lot and a whole lot of hard hats. But by October, the plan is for this to have a grand opening and reopening. And Precision is in this, ELO is going to be in the second floor of this building. All of this will be us, and very exciting times for us at this point. And then I also wanted to announce that we formed this entity only less than a month ago, and we already have a subsidiary in Australia. So this is in collaboration with QUT, with Saga, who is here in the audience. We have a plant-based proteins program that we are advancing with QUT here. And ELO will be located on the QUT campus in Brisbane. So with that, I just wanted to put in a quick plug here. We are hiring. Did I say that right? We are hiring, and please reach out to me. We are hiring at all levels. It's a great place to work. RTP, a lot of things happening at RTP, and great time to be a part of a growing company. Thank you.